introduce the next speaker. This is my boss, Peter Libby, who, as you know, has been an outstanding authority in the area of atherosclerosis, inflammation, and as the chief of the division, Peter is also the Mellon-Pratt Professor of Medicine, Harvard Medical School. Peter.
Thank you very much. So it's my job to give you some insight into how this disease works at the basic level. Veins are different from arteries. They have a structure which is a bit more amorphous on the outside compared to a little artery here, but their insides are much more complex. As you see here, they have veins, structures which we don't like to see in arteries. We've already heard about Rudolf Virchow and Virchow's Triad. This is Virchow's Anatomical Museum at the Charité in Berlin. This is actually Virchow's lecture hall in a state of disrepair. I guess you could call this extreme deferred maintenance. There's Virchow's podium there, and here it is today. They call it Die Ruinen in Berlin. And in the Anatomical Museum, here in the section called Thrombose, you see these arterial specimens with thrombi in them. So you can today have the legacy of the history that we've already heard about from Dr. Sasahara. And Dr. Sasahara already introduced to you the concept of Virchow's Triad, venous stasis, vascular injury, hypercoagulability. And as you will hear played out through these talks, there are a number of factors which are associated with each limb of this triangle. Mobilization, heart failure and low flow, venous insufficiency for venous stasis, obesity, cancer, infection and the acute phase response for hypercoagulability, along with pregnancy. And in terms of vascular injury, prior deep venous thrombosis or pulmonary embolism, fractures, surgery, or other kinds of trauma. So this is really where we were in the 19th century and with the accumulated clinical experience of the ensuing decades. But we've now entered a new era, the era of vascular biology. And we possess tools that Virchow actually didn't have. He had the microscope, he had his keen sense of observation and marvelous brain, but he didn't have electron microscopy or any of our modern cellular biological tools or molecular tools. This is what the endothelium of the vein looks like if you look down on it from the perspective of a blood cell. And these structures here are the endothelial cells, the cells that line every millimeter of our vasculature. And the story of the vascular biology of venous thrombosis is very much a story of this cell, something that Virchow appreciated. He entitled his textbook Cellular Pathology, but didn't have the tools to dissect with the depth that we have today. Now here in cross-section, in a transmission electron microscope, is the endothelial cell, the hero of my story, overlying a basement membrane. This is the middle part of the vein with a few vascular smooth muscle cells, venous smooth muscle cells, and then the outside of the vein is shown here. So how is the endothelium implicated in the biology of this process? The normal endothelium is really a remarkable surface. It is the only surface, either man-made or synthetic or natural, that can maintain blood in a liquid state during prolonged contact. The normal endothelium is anti-inflammatory, vasodilator, that is, it keeps the channels open, and very importantly for our discussions this afternoon, possesses anti-clotting mechanisms, anti-thrombotic mechanisms, and what we call profibrinolytic mechanisms. The drugs, which we heard about from Dr. Sasahara, are actually extensions of our own natural clot-busting system that we each have in our own bodies. And the normal endothelial cell supports the fibrinolysis. So if a stray thrombus should form, we have our own internal sentinels that will try to lyse that clot. So here is the nice normal endothelial cell with all of its homeostatic desirable functions, experiencing normal flow and having low thrombogenicity and high fibrinolysis, so less tendency to form a clot, and should a clot form, a high tendency to chop it up and break it down. What are the anticoagulant mechanisms that are associated with the endothelial cell? There's a small molecule which is made by endothelium known as prostacyclin that is a very potent vasodilator. It expands the vessel, and it also combats thrombosis by inhibiting platelets, which are the formed elements in blood, which are very important in propagating thrombi. The normal endothelial cell also makes a gas called nitric oxide, recognized a few years back with the Nobel Prize, which also is vasodilator and combats the aggregation of blood platelets. The normal endothelial cell has on its surface a special molecule called thrombomodulin, which very curiously takes thrombin, which is thought of as the key molecule in generating clots by processing fibrinogen to fibrin, and turns this usually procoagulant molecule into an anticoagulant molecule by activating a series of other proteins that are downstream. And the endothelial cell has this particular magical property, if you will, of turning the procoagulant thrombin in low doses to an anti-thrombotic. You heard about heparin. Heparin is not just found in the drugstore shelf, but also is made by the endothelial cell in an endogenous form, heparan sulfate proteoglycans, which line the coating of this marvelous endothelial monolayer. And then, as I told you, the endogenous clot busters, urokinase, which we heard about from Dr. Sesahara, and tissue plasminogen activator are products of the normal endothelial cell. So that's the normal homeostatic state that we hope all of our endothelium is in, both arterial and venous. But unfortunately, there's the evil side of the endothelial cell. When it gets hot and bothered, it changes its face to one that, instead of combating thrombosis and promoting fibrinolysis, does the opposite. And we have a great deal of molecular understanding now of how that works. Some of the ways in which the activated endothelial cell can change its face in terms of clotting is by augmenting the release of inhibitors of fibrinolysis. So we have our endogenous clot busters, but we also have the inhibitor. And the endothelial cell can secrete in high quantities an inhibitor known as plasminogen activator inhibitor, PI1 for short, that is able to bind the endogenous lytics and stop their usually salutary action as sort of a sanitation system to clear up any thrombi that should form. The endothelial cell, when it is in an activated state, will reduce the availability of this gas which combats thrombosis, and will also rev up its production of a molecule known as tissue factor, which is a very potent procoagulant which turns on the blood clotting cascade by activation of factor 7 and factor 10. Now here in cartoon form is what I've told you in some words. This is the generation of plasmin, which breaks down fibrin, which is the major protein in clots, which is generated by the action of thrombin from fibrinogen. Plasmin comes from a precursor known as plasminogen, and the plasminogen activators that we just heard about actually turn plasminogen on so that it can make plasmin and cause the breakdown of clot. But as I told you, this inhibitor can bind to these endogenous lytics and block this very important mechanism for clearing clots that are formed. So the endothelial cell has, associated with its surface or as secretory products, a whole gamut of regulators of thrombosis and clot stability. I've been through each of these, and with the exception, perhaps, of von Willebrand. In fact, I see Lisa Wagner in the audience, who's one of the world's experts on this very important clotting factor, potentiator of blood clotting. So, with all of those different capacities, in the normal endothelial cell, the one with the happy face, the teeter-totter is in this direction. But when you have the endothelial cell activated and angry, it switches its character, and the teeter-totter balances in the other direction. So what are the triggers for this activation of endothelial cells? So here's where we now can go back to the clinical and pathological experience of the decade and try to integrate it with this concept of the primacy of the endothelium in regulating venous thrombosis. Mechanical injury, obviously, if the endothelial cell isn't there, it can't exert its hygienic, helpful functions. We know that inflammation and proteins that are important mediators of inflammation that we call cytokines are key regulators of these properties of the endothelial cell. And when pro-inflammatory cytokines impinge on the endothelial cell, it will tip that teeter-totter in the adverse direction. There are acute phase reactants, so that when we have a systemic inflammatory response, let's say caused by an infection or overwhelming injury, the liver will pour out excess fibrinogen, which is the precursor of clot, and will also, not just at the level of the endothelial cell, but systemically, turn on the production of that inhibitor of our endogenous clot-busting system. We have this one little bullet here encompasses a whole realm of wonderful basic science that we really understand in precise molecular detail, down to various angstroms of resolution of the X-ray crystallographic appearance of the players. But just to convey to you very simplistically, when one has an infection, let's say with a gram-negative bacteria, very common in urinary tract infections, for example, or in bowel infections, the bacteria have endotoxins that are lipopolysaccharides, we call them LPS. And they can turn on a whole cascade of pro-inflammatory mediators that will in turn flip the switch on the endothelial cell, taking it from its normal homeostatic state into its procoagulant and anticoagulant state. And then there's cancer, a very strong association between venous thromboembolism and cancer, and recurrent venous thromboembolism. And we now have a very good handle on one of the major molecular mechanisms of this association between cancer and thromboembolism, and that is the generation by cancer cells of microparticles that they throw off that contain that potent procoagulant tissue factor. So the cancer will secrete these tissue factor-bearing microparticles. There's an X-ray crystallographic structure of tissue factor, and there are a number of laboratories that have very nicely shown the association between cancer cells and heightened tissue factor, including the laboratory of our neighbor, Bruce and Barbara Curie, to speak, who had a very nice masterful review in the New England Journal very recently about this general concept. Now, I mentioned that flow is a very important regulator of the endothelial cell. You know, we think that stress is bad, but for the endothelial cell, laminar shear stress here is actually a good thing. It keeps the endothelial cell in its protective state. So how is it that flow can regulate the endothelial cell? What's the link between the biomechanical stimulus of laminar flow, nice regular flow, like a peaceful growth, and normal function of the endothelial cell? Well, this is a nice Brigham story. It turns out that there is a transcription factor, a master factor that regulates gene expression at the level of the cell nucleus that has the uneuphonious name of purple-like factor 2, KLF2, that regulates endothelial hemostatic functions. And this is a very nice Brigham story of collaboration between departments from cardiology and pathology, and they have identified this KLF2 as a master regulator of this thrombolytic and anticoagulant kind of property of normal endothelium. Overexpression of this transcription factor strongly induced thrombomodulin, endothelial nitric oxide, and reduced the inhibitor of endogenous fibrinolysis. The way that that works is by robbing Peter to pay Paul. So here in the cell nucleus, if you will, there is the evil twin of KLF2, which we call nuclear factor kappa B, and that is a master regulator of bad guy proteins, the genes that encode bad guy proteins, including the pro-inflammatory cytokines. And it turns out that KLF2 and NF-kappa B share some important co-stimulatory factors. So when normal flow turns on KLF2, it robs the NF-kappa B of the cofactor that it needs to turn on the bad guys, and so you not only turn off the bad guys, but you turn on the good guys. And it's interesting to keep in mind for future reference that the statin drugs, which we think of as cholesterol-lowering drugs, also turn on this system. Stay tuned for breaking evidence that will be presented by Dr. Richter at the American College of Cardiology about links, potential links between statins and venous thromboembolism as well. So this is where we are, Virchow's triad, the risk factors that we put here. But unfortunately, I didn't know Art, that he was known as Dr. Rainer Virchow, but he is rather slim himself, as you see here in this artistic depiction. And in fact, the population in Berlin in his day had a body mass index a lot lower than those who are walking the malls and streets of America today. Actually, they aren't walking. That's the problem. So I think we need to revise Virchow's triad. And we need to think more of a quartet. Because the risk factor, which is burgeoning our society, and any casual observer knows this, is obesity and diabetes. We have growing obesity, growing risk of diabetes. And it turns out that I think looking forward, unless we can stem this epidemic, this is going to be a major source of triggers for venous thromboembolism. Why? Many, many laboratories, just one example, have shown that in diabetes, that inhibitor of our endogenous clot busters is increasing. And here's a protocol where patients underwent an infusion of some fat in a soluble form and sugar to mimic the diabetic state, the uncontrolled diabetic state, and you see that you rev up the production of this inhibitor of our endogenous clot busters. So obesity increases this inhibitor of our endogenous clot busters, and that causes the clots to accumulate in kidney form. And it turns out that the endothelium is a master regulator of many of the adverse effects of diabetes and its metabolic consequences on not just thrombosis, but also inflammation, which, as I'll close with in a moment, is upstream of thrombosis very often, and also the caliber of vessels, which is a critical aspect of arterial and venous diseases. So to close, I just want to share with you my worldview, which is that inflammation is a common theme in many of our diseases, chronic and acute, and that inflammation can beget thrombosis, and thrombosis can beget inflammation, which is really more recently appreciated. My former colleague, Dan Simon, and I wrote an editorial about this a few years ago, Inflammation and Thrombosis, the Clot Techniques. And the updated story looks something like this. Here's thrombin, which we think of as an activator of clot formation. But it also can activate some of those muscle cells in the walls of vessels to migrate and proliferate. It can cause platelets to activate and release a bunch of pro-inflammatory mediators, and can also be a major trigger for shifting the functions of the endothelial cells from those normal homeostatic ones to those which will promote thrombosis. So inflammation is inextricably linked with the adverse functions of the endothelial cell, which we think are at the root of venous thromboembolism. So it's wonderful to have you here and to be able to share with you how we can move from the basic science laboratory through pathology and medicine for large clinical trials to try to provide the best in our